just now, and then the rest of the day for contemplation or whatever else you want to do. And so the first speaker is somewhat of a legend in the antibody engineering field. Is Tomoyuki, who's the head of research development at Chugai, and he's a world leader in efforts to develop novel antibody engineering strategies such as bispecifics, half-life extension, recycling, antibody technology. And I'm sure there's nobody in this room who doesn't know of Tomo. So, Tomo.
No? Okay. Oh. Okay. Dennis, thank you for very kind introduction. And thank you, Fair Journey Biologics, for giving me this very unusual opportunity to speak on the boat. My presentation will be 45 minutes, but since we are on the boat, you cannot escape from my presentation. And I think I cannot also escape from your questions. Okay, so today I would like to talk about the design of antibody therapeutics with engineered specificity. So let me first very briefly explain who we are. So our company is Chugai Pharmaceutical. We are a Japanese company located in Tokyo. So we are a member of Roche Group. However, we maintain an autonomous operation. We have a very independent research and early development. Although we collaborate with Roche and Genentech, but we have a completely independent research. So Chugai has been working for antibodies for more than 20 years. Our first product was actually Actemra or tocilizumab, which is IL-6 receptor antibody. Including Actemra, so Chugai has achieved 9 FDA breakthrough designation, including 4 antibody products. So first one was the Actemra. The second one was Hemlibra. So this was the first human IgG bispecific antibody against factor 9A and factor 10 for the treatment of hemophilia. The third one was the Enspring in 2020. So this is the first recycling antibody that was approved. Last year, we got breakthrough designation for IL-31 receptor antibody nemolizumab, which is used for pruritus. So we have been working on, especially focused on engineered antibodies, and today I will be talking about these 3 topics. So I would like to focus on how we can engineer the specificity. So the first one is a switch antibody. So this has been already published, so I will not use a lot of time on this. So this is an extracellular ATP adenosine triphosphate dependent antigen binding. So this allows us to conditionally activate antibody where the extracellular ATP is in high concentration, which is the tumor microenvironment. The second and third, I would like to use most of my time in my presentation, so these are unpublished. So the second one is called DualIG. So this is a next generation T-cell bispecific antibody with an improved efficacy and safety profile. So we engineered the CD3 portion of the T-cell bispecifics so that it binds not only to CD3, but also binds to CD137, which allows us to give signal 1 and signal 2. The last one is quite interesting antibody. So this is a multi-specific antibody. So this is an antibody for treatment of celiac disease. So celiac disease is caused by a multiple gluten peptide, which is presented on MHC. So our antibody recognizes more than 25 different gluten-derived peptides, which has very different sequences. So I will show you how we generated this antibody and how this antibody recognizes these multiple different peptides in a structure using the crystal structure. Okay, so let me start with the switch antibody. Okay, so before moving on to switch antibody, so we have been working a lot on engineering environment-dependent binding antibody. So the first one was a pH-dependent binding antibody. We call it recycling antibody. So this antibody has a reduced antigen binding at acidic environment, so resulting in a pH-dependent interaction. So we generate this by introducing histidine substitution in the variable region. So we have several products that we apply this technology. So after this, we noticed that not just pH, but also the calcium ion concentration is reduced in the recycling endosome. So we made an antibody that has binding dependent on the calcium ion concentration. So to make this kind of antibody, we found that it is very useful to have a library that has a calcium binding motif in the CDR, so that the binder will always have a calcium-dependent binding. So we successfully generated this calcium-dependent binding antibody with this phage library having a calcium-dependent motif in the CDR. Then we noticed that we can do more. So what we did is we made an antibody that only binds to the target in the presence of adenosine triphosphate ATP. So this antibody has an ATP binding pocket in the CDR. So therefore, the antibody can only bind to the target only in the presence of ATP. So I will have a bit of detailed presentation on the switch antibody first. Yeah, okay. So first, we wanted to make an antibody tumor specific. So many of the cancer immunotherapy or antibody drug conjugates suffer from on-target off-tumor toxicity. So therefore, we wanted to make antibody more specific to the tumor microenvironment. So we found that the extracellular ATP is elevated in the tumor microenvironment. So the left figure is the right figure is the mouse study showing that the extracellular ATP is elevated. The middle figure shows that in human, extracellular ATP is elevated. So actually, the extracellular ATP concentration in a normal tissue or in plasma is tightly regulated, and the concentration is around 30 nanomolar, very low. On the other hand, within the solid tumor microenvironment, the high concentration intracellular ATP is released from the dying or stressed cancer cells, resulting in more than 100 micromolar extracellular ATP concentration. So there is almost 1,000-fold difference in the extracellular ATP concentration, and we thought that we could use this extracellular ATP as a switch to turn on the binding of the antibodies. So the concept is described here. In the high ATP concentration, the antibody can bind to the antigen, but in the low ATP concentration, such as normal tissue, the antibody cannot bind to the antigen. So how we obtain this kind of ATP-dependent antibody? So what we did first is we first obtained an antibody against ATP. We used a rabbit immunization and obtained an ATP binder. And among the various ATP binder, we selected one of the clone that has a deep pocket in the VHVL interface, so that ATP is like buried in the VHVL interface. So this is the clone that we used. We solved the crystal structure, and we identified the residues where it is not involved in ATP binding. So the pink areas described here are the residues that is not so much involved in ATP binding. So therefore, we diversified this position in the phage library. So we made a synthetic library having a diversity in these pink regions. So this library maintains the ATP binding capability so that if you do a phage panning in the presence of ATP and also do the negative screening in the absence of ATP, you can obtain the ATP-dependent binder. So this is one of the first ATP switch antibody we obtained. The antigen was IL-6 receptor at that time. So you can see the crystal structure here that ATP is actually sandwiched in between the antibody and the antigen. So this allows the antibody bind only as an ATP antibody complex. So in the absence of ATP, this antibody cannot bind to the IL-6 receptor. In other example, so this is a switch antibody against PD-1. So this is a crystal structure with or without ATP. So you can see that the VH, maybe there is no structural difference in the presence or absence of ATP. But if you look at the light chain CDR, especially light chain CDR3, the presence of ATP induces a structure change of the light chain CDR3 that allows the PD-1 recognition. So this antibody also has a very strict ATP dependency. So we applied this ATP switch technology for 4-1BB or CD137 agonist as a first project. As you know, the CD137 agonist has been tried by various companies, but because of the systemic toxicities, mainly the liver toxicity caused, due to the liver toxicities, the dosage cannot be increased. So we decided to make an ATP switch CD137 agonist antibody. So we obtained the antibody that binds to human CD137 in ATP-dependent manner. So the upper figure shows the biochar sensogram. You can see the ATP-dependent binding. The bottom figure shows the in vitro assay, interferon gamma production by CD8 T cells by our antibody. It's concentration dependent, and in the absence of ATP, there is no interferon gamma production. So we confirmed in vitro that this is an ATP-dependent CD137 agonist. So this is one of the in vivo studies that we conducted. So this is human CD137 knock-in mice grafted with a colon 38 mouse tumor. So STA551 is our switch antibody, and we compared with a non-switch antibody. So we use relumab as a representative of a non-switch antibody. So the tumor volume change in the left figure, we can observe almost similar efficacy. That's STA551 and non-switch shows similar efficacy. On the right figure, we checked the immune activation status in the liver. So the y-axis shows the PD-1 expression on CD8 T cells in the liver. So if you look at the pink one, non-switch, you see a dose-dependent T cell activation. And you can see the activation even at the lowest dosage, 0.093 milligram per kg. But if you look at STA551, which is a switch antibody, we don't see any T cell activation even at the highest dosage. We also checked other organs such as spleen, lung, and colon, and we consistently observed no or very residual T cell activation in the normal tissue. We conducted a monkey GLP tox study of this STA551, and it was tolerated up to 500 milligram per kg. And with those results, we started clinical development of this molecule, and now we are testing this molecule in combination with atezolizumab in phase 1 study. So the next one is the dual IG. So then I will talk. So first I will talk about engineering the bispecifics and beyond. So Chugai first started to work on bispecific antibody in from about 2005 or something, when we had the idea of mimicking factor 8 using factor 9A, factor 10 bispecific antibody. So we generated several bispecific antibody technologies called ART-IG and FAST-IG. So this allows us to bind to 2 different antigens. The light chain pairing issues was first solved by common light chain, and then we established a method of controlled chain pairing. So emicizumab was our first bispecific antibody product, and we have a next 007. So this is a next generation emicizumab product, which is more potent than emicizumab and which is more have a longer half-life than emicizumab. So this is now tested in phase 1/2 study. And once we developed this bispecific antibody, we started T-cell, so-called T-cell bispecific. So we have a Gripkin-3 CD3 bispecific in phase 1. So after we established this 1+1 type of bispecific antibody, we started to work on FAB with a dual specificity. So as you can see here, the figure, the single FAB binds to antigen A and B with an overlapping epitope. So therefore, antigen A and antigen B would compete each other. So we discussed what kind of thing we can apply this kind of technology. And we found that creating a second generation T-cell bispecific using this dual IG would be an attractive approach. So we started this project. So the last one is a multispecific, so I will talk about this later. So let me talk about this dual IG. Okay. So I don't think I need to explain this. So this is a common T-cell bispecific. So one arm binding to the tumor antigen, and the other arm binds to the CD3. And by cross-linking, it activates T cells, and T cell kills the cancer cell. So this is one of our molecule, LE974, which shows quite good efficacy in this model. And this is now also in phase 1. However, this T-cell bispecific has a lot of problem. So one of the issue is that we can observe high efficacy where the tumor is rich into T cell. So this is a publication from, I think, Regeneron. So if you look at the right figure, with a high T cell infiltration, the T cell bispecific can show good efficacy. But if the T cell infiltration is very low, we don't see efficacy. So we wanted to solve this issue. So we decided to work on CD137 because, first of all, for the CAR T cells, CAR T with only the signal 1 was not sufficient, even in the hematological cancer, but the CAR T cells with the signal 1 and signal 2, such as CD137 or CD28, turns out to be very efficacious in the CAR T cells. And we thought CD137 is better because CD28 may be too dangerous. So our antibody, the T cell binding arm, binds to both CD3 and CD137. Therefore, this dual IG, in addition to CD3-mediated T cell activation by signal 1, this dual IG can induce a co-stimulatory signal, which is signal 2, by also cross-linking CD137 only in the presence of tumor antigen, like this. So one of the important points of this dual IG is that the dual IG is designed to bind to both CD3 and CD137, but not simultaneously. So this is very important because if the antibody can bind to CD3 and CD137 at the same time, these T cells can kill CD137 expressing immune cells like NK cells or whatever. So this will lead to non-tumor antigen dependent T cell activation, which is not good. So we designed our molecule so that CD3 and CD137 will never bind at the same time. So how we did this? So we did similar approach as we did for the ATP switch antibody. So first, we had our anti-CD3 antibody recognizing the N-terminal of the CD3. So this is the crystal structure. So we determined the position which is not so much involved in CD3 binding. So those are the pink residues. So these pink residues are diversified in the phage display synthetic library. So then we did the phage panning against both CD3 and CD137 so that the FAB will bind, so that we can find the clone that recognizes both CD3 and CD137. So once we identify a lead antibody which can bind to both CD3 and CD137, then we moved on to the optimization process. So this is one of the platform technology that Chugai applies. So we have this comprehensive mutagenesis method. So we can make these all CDR residues substituted to other 18 amino acids other than the cysteine. So these more than 1,000 variants we can produce in a purified protein within 2 weeks or so. So with this rapid protein production and screening, we can this platform allows us to scan all the mutation in the CDR residues and find advantageous mutation. So then we conduct a combinatorial mutagenesis, and we repeat this process multiple times. And finally, we obtain a clinical candidate. So in this dual IG case, we conducted multiple rounds of optimization and identified several dual binder. The dual binder means the antibody that binds to both CD3 and CD137. So then the question was, what is the optimum binding kinetics to CD3 and CD137? Is it good to bind to CD3 and CD137, both high affinity, or maybe either one should be low affinity, or KO, does the KO and KO matters? So we made a various variants of dual binding FAB with a different CD3 and CD137 kinetics. So these are some of the biochrosensogram of the different clones. Then we conducted some in vitro assay to find out what is the best. So this is the IL-2 production and interferon gamma production of the different variants with different binding kinetics. So the red one is the conventional T-cell bispecific with just CD3 binding. So we found that the antibodies with different kinetics have different cytokine production profile. And among them, we determined that this green clone is the best. So if you look at the sensorgram, binding kinetics of this green clone as compared to the conventional T-cell bispecific, we can see that the CD3 binding is much weak. The off rate is very fast. So this CD3 is, I think the KD value is like a 30 nanomolar, but this CD3 is like 100 nanomolar or more. So very, very weak. But we have a quite strong binding affinity to CD137. So we found that this clone shows the best property, both in vivo and in vitro and in vivo. So one of the important points of this weak CD3 binding is that if the CD3 binding is too strong, it cannot bind. So CD137 expression requires CD3 signaling. So once the T cell receives CD3 signaling, T cell will express CD137. So if the antibody binds to CD3 with too strong affinity, it cannot dissociate from CD3 to bind to CD137. So this fast K-off was quite important so that the antibody first binds to CD3, induce CD137 expression, and then binds to CD137 for signal 2. And this high affinity was required for strong CD137 signaling. And this weak CD3 affinity was also important for cytokine release syndrome. So I think it is known that the T cell bispecific have this CRS IL-6 production issue, and this IL-6 production clearly correlate with the CD3 affinity. So if you have an antibody with very high CD3 affinity, a lot of IL-6 is produced. So this molecule has a weaker CD3 affinity, so the IL-6 production is actually smaller than the T cell bispecific. So what we did first is the in vitro assay to confirm that this dual IG can really induce proliferation of the T cell. And also one of the concepts was the CD137 signaling can prevent or rescue the T cell exhaustion. So this right figure shows the scheme of this in vitro assay. So this is like a long-term in vitro incubation culture assay. So either so TRAB is CD3 bispecific. So CD3 bispecific or the dual IG is incubated for 7 days. Then we recover the T cells, and then we incubate again with either TRAB or dual IG. And after 9 days, we check the cytokine production, and also we check the T cell proliferative activity. So you can see here that if you incubate with TRAB and then switch with the TRAB, so CD3 only, after 9 days of incubation, the T cells are exhausted and no interferon gamma production. But if you start with dual IG and incubate with dual IG, so we see a very strong interferon gamma production. And after incubating with TRAB and then switch to dual IG, we also see interferon gamma production. So we thought that with this, the dual IG have a potential to prevent and also rescue the exhausted T cell. Yeah, and for T cell activation, similar, so dual IG and dual IG shows the best proliferative activity. So the target that we applied this dual IG technology first is the DLL3. So DLL3 has been pursued by Amgen BiTE platform and have shown quite good clinical efficacy. So DLL3 is a very clean target, so that we thought that it might be a good idea to apply this technology to DLL3 first. So this is a Hunog mouse grafted with a lung cancer cell line expressing DLL3. So this cell line is extremely immune desert lung cancer cell line. So you can see that the number of T cells in the tumor is very low. So it is like only several T cells, 3 or 4 or something, less than 10 T cells per milligram of tumor. So I think this is very immune desert model. So in this model, the simple T cell bispecific with CD3, the red one, shows a modest efficacy. And this is probably due to the number of T cells, although it increased to some extent at day 7, but it rapidly decreased at day 14 and day 21. But if you treat this tumor with a dual IG, the number of T cells increased by like nearly 1,000-fold, and the number of T cells reaches more than 1,000 per milligram. So this is quite similar to the number of CD8 T cells in the blood. So the T cell bispecific works well in the hematological cancer because there's a lot of T cells. So we believe that with this dual IG, since we can increase the number of T cells in the solid tumor to a similar level as a hematological cancer, we hope that this molecule will be also effective in patients. So this molecule is now being tested in phase 1 study. So this is the second target that we are working on. So sorry that I cannot disclose the target. So this is a different model using a human CD3, human CD137 knock-in mice grafted with a mouse tumor with a target X transfectant. So you can see better efficacy, anti-tumor efficacy, higher interferon gamma production, and higher CD8 T cell infiltration. Then we checked the safety profile. So we injected the conventional T cell bispecific TRAB, this one, and dual IG at different dose level. So I was asked that I cannot disclose the exact dosage. So dosage A is the lowest level. So for the conventional TRAB, at the dose level of 100, higher than this dose level A, we see a very strong IL-6 production. And if you look at the in-life finding, you see a lot of changes, and this was not tolerable. In case of JORIG, we see almost no IL-6 production, even at this 1,000-fold higher dosage. And in life finding, it was very much well tolerated. So this difference comes from the low CD3 binding affinity for JORIG. TRAB has 30 nanomolar CD3 affinity that induces this kind of high-level IL-6 production, but JORIG with lower CD3 affinity, it was tolerated at least up to this level. Okay, so then I will move on to the structural point of this molecule. So because of the nature of our phage library, which was used to obtain this JORIG, you can actually see that this CD3 and CD137 is recognized by antibody using this overlapping paratope. So because this is overlapping paratope, these two molecules, CD3 and CD137, cannot bind to the antibody at the same time. And it was, so if you look at this peptide sequence, right here and right here, the sequence is very different. But nevertheless, the antibody can recognize these two different sequences. So we thought why this happens. So we looked into detail of the structure. First, so if you look at the heavy chain CDR3, there is a huge conformational change when it is bound to CD3 or CD137. So the heavy chain CDR3 takes a different conformation when bound to CD3 and 137. And we found that the several different CDR residues in the JORIG recognize solely either CD3 or CD137. So these are heavy chain CDR3 residues. And you can see that, for example, position 102, histidine, so this residue recognizes only CD3. 104 and 105, it recognizes only CD137. So these residues are recognizing either CD3 or CD137. On the other hand, there are several residues that the same residues recognize both CD3 and CD137 in a different manner. So especially if you look at this tyrosine 103 right here, so when this tyrosine is recognizing CD3, it is facing this way. But when this tyrosine 103 is recognizing CD137, it is like facing downward. So we thought this is quite interesting. So tyrosine has a multiple recognition function. It has a hydroxy group, it has aromatic group, so it can have a multiple interaction. So we thought that tyrosine can be like a very good residue to allow us to engineer this multi-specific binding. And we found that in the next celiac disease project, tyrosine plays a very important role in the multi-specific interaction. Okay, so I have 12 more minutes. Okay, so last talk is this celiac disease antibody. So yes, so this antibody binds to the multiple target. Okay, so as you may know, the celiac disease, there is no therapeutics other than the gluten-free diet. So there is a huge unmet medical needs, but no treatment. So it is known that the celiac disease is caused by gluten peptide HLA DQ2.5 T cell interaction. So more than I think 90% of the celiac disease patient has HLA DQ2.5. This is because that the gluten peptide is only presented on DQ2.5. However, there are various gluten peptides more than 25 gluten peptide, which is known to induce this pathogenic T cell activation. So we need to block all these gluten peptide activating the T cells. So the therapeutic concept is that the antibody that selectively binds to multiple various gluten peptide, but does not bind to DQ2.5, presenting irrelevant peptide. So we did not want to block all the DQ2.5 because it will probably lead to a safety issue. And DQ2.5 is abundantly expressed in the body, so the PK would be very poor. And the DQ2.5 is expressed on antigen-presenting cell, so probably the immunogenicity would be very high. So we wanted to have an antibody that only binds to DQ2.5, expressing this gluten peptide. However, this was not easy because these are the reported gluten peptide sequence in the celiac disease. So if you look at the sequence, you can see multiple proline and glutamic acid and glutamine, but sequence is very different. And the 5 peptide, these are the alpha gliadins and the omega gliadins. These are known to be the 5 immunodominant peptide. So this is very pathogenic peptide. But even among this most pathogenic gluten peptide, the sequence homology was like 33 or 78%, very different. However, we wanted to make an antibody that recognizes all these peptides in complex with DQ2.5. So we started with rabbit immunization. So the immunogen that we used is this 33-mer gliadin peptide. So this 33-mer gliadin is a unique peptide in the gluten that contains multiple immunodominant epitopes like this and this and this. So we thought that if we immunize the rabbit with this peptide, we might obtain cross-reactive binders against different immunodominant peptide. So we immunized 33-mer conjugated or linked with HLA-DQ2.5. We use this as an immunogen, and we did B cell screening method. So rabbit was immunized with that immunogen, and we sorted out the B cells, and we did supernatant assay, and we did the gene cloning, and we did a lot of screening. And then we identified 2 very unique clones called 385 and 344. So we thought that these 2 are quite unique and these are complementary. So we checked the binding property to these gluten peptide, and we found that 344 clone and 385 clone is complementary recognizing the different peptide. So 344 recognizes these, but it cannot recognize this one. But on the other hand, 385 can quite strongly recognize this peptide. So we decided to make a bispecific, although it's a multispecific, but a bispecific IgG format with 344 in one arm and 385 in the other arm. Then we went on to multi-dimensional optimization. So we did the optimization using our comprehensive mutagenesis and combination strategy. So finally, we were able to identify a clinical candidate called DONK52. So with one arm, 385, and one arm from 344. And we applied our half-life extension technology. So this is the binding profile of our clinical candidate, DONK52. So you can see that the DONK52 does not bind to other HLA-DQ. It doesn't bind to DQ2.2 and other DQs. It doesn't bind to DQ2.5, presenting irrelevant peptide like this. And it binds to all these different peptide presented on HLA-DQ of 2.5. So we used as a pan-DQ as a comparator. So this middle one. So this is recognizing just the DQ 2.5. So it binds to all the DQs and DQ 2.5 presenting irrelevant peptide. So then we conducted several non-clinical studies. So this is the inhibition, in vitro blockade of T cell activation. So this is, we established the assay of mixing B cells and Jurkat cells expressing DQ 2.5 and T cell receptor recognizing different these peptide. So you can see that DONQ52 can inhibit the T cell activation in dose-dependent manner for all these different peptides. And also this pan-DQ, so this is a control, it can also inhibit. And we conducted an in vivo study using a HLA-DQ 2.5 transgenic mice. So this mice was immunized with either 33-mer gliadin, so this is a peptide we use for immunization, or KLH, keyhole lymphocyte hemocyanin. So, and we did, we recovered the splenocyte and checked the T cell activation. So first, if you look at the immunization of the 33-mer gliadin, both the pan-HLA-DQ antibody and DONQ52 can both block the T cell activation. If you look at the T cell activation after immunizing KLH, pan-DQ, because this is a pan antibody, it inhibits T cell activation. However, DONQ52, because this is specific to gluten peptide, it doesn't inhibit the T cell activation mediated by KLH. So with this result, we can say that the DONQ52 can suppress T cell activation only against the gluten peptide. And we also checked the PK of these two antibodies, because as I mentioned, DQ 2.5 is highly expressed over the body. So you can see that the pharmacokinetic of pan-HLA-DQ is very poor. But on the other hand, because our DONQ52 is only specific for gluten peptide complex DQ 2.5, we see a very long half-life. So with this long half-life, we expect that we can dose this antibody by subcutaneous injection for chronic use for the celiac disease patient. So now this molecule is now tested in phase 1 study. So I will explain in the next 4 or 5 slides why our antibody recognizes this multiple peptide. So this is a crystal structure of the 344 arm and 385 arm. And we have 2 structures for each arm with a different peptide. So the recognition is like a TCR. So this is like a TCR-like antibody, recognizing the DQ 2.5 from the top. And you can see that it can recognize these 2 different peptides. So it has protein, common protein, but these residues are very different. Nevertheless, this antibody can recognize. So why this can happen? So first, our antibody is specific to DQ 2.5 because some of the CDR residues in both arms recognize the unique residues for the unique residues to HLA-DQ 2.5. So these are the residues which are unique to DQ 2.5. So therefore, both antibodies are DQ 2.5 specific. And the next is why it can recognize very different sequence. So we found that this protein and glutamine-rich motif of gluten epitopes. So these epitopes are known to be very critical for the pathogenesis. So all the pathogenic gluten peptides contain this protein glutamic acid, glutamine-rich motif. And probably our antibody recognized this motif. And this recognition was mediated by multiple tyrosine residues. So you can see, so if you look at the serial sequence of our antibody, there are a lot of tyrosines, much more than the normal antibodies. And if you look at the tyrosines, that this tyrosine plays a very important role in recognition of all these peptides. And when it recognizes different peptide, it faces different way. So therefore, this very flexible recognition by tyrosine is probably playing an important role in recognizing very different peptide. Okay, so in summary, so we were able to design antibody with engineered specificity, switch antibody with ATP dependency, dual Ig, which can bind to both CD3 and CD137, and the multi-specific antibody recognizing more than 25 different gluten peptides. So this is my last slide. So we have a lab in Japan and Singapore, and we are, and we have so many scientists contributed to this work. So I would like to acknowledge them. And today in this boat, we have two colleagues joining, so Taichi and Hitoshi. So they are joining this conference. So if you have any question, you can also ask them. So that's all. Thank you very much.
So I'm interested in your dual Ig antibody CD3, CD137. So if you do a primary screen, and I assume that you do sequential panning, so what do you typically get out of a screen like that? Are these very rare events? Do you find quite a few diverse clones? And how do they, what are the affinities for the two different targets, just in the primary screen? Because it's, it's a bit unusual.
So the dual Ig CD3 and CD137. Yeah. So the initial lead binders are very low. So the binding to CD3 was like a micromolar, and binding to CD137 was also in the micromolar range. So we had to affinity maturate for both binding.
And rare, I mean, just a few different sequences, or is this a panel that you're getting?
Yeah, we found multiple sequence. Yeah, but the sequence of multiple, but because it recognized the same epitope. So actually, the diversity of the clone was limited, I think. The sequence was diverse, but probably all the clones are recognizing the same epitope.
And then you have to do a lot of engineering to get to the affinities that functionally make sense.
Yes.
Okay.
Yeah.
Thank you so much for your presentation. I'm wondering what kind of range of affinities did you get with multi-specifics against different peptides?
Yeah. So the red peptide, the affinity is like single-digit nanomolar, and this like light colored one is like 10, 30 nanomolar. So, but still, it all are in the nanomolar range.
Thank you.
Hi, thanks so much for your talk. Fascinating and very impressive. For the CD137 co-binder, does it compete with the CD137 ligands or allow binding for that ligand?
Fantastic talk and fantastic engineering, protein engineering work. Just wondering for your dual IG, you're using the same molecule to bind both CD3 and 4-1BB, but not at the same time. I'm wondering if comparing to using two molecules, one molecule against CD3 and the other molecule with anti-4-1BB, what's the, you're comparing your dual IG with TRAP-B. But what's the advantage of one molecule over two molecules in this? Just
Okay, so yeah, that's the question we frequently receive. So we can probably achieve similar efficacy if we combine CD3 tumor antigen bispecific plus CD137 tumor antigen bispecific. So in terms of safety, no, efficacy may be similar. Yeah, we did the study and it was similar, actually similar. So one of the important thing is, so our dual IG, the CD3 binding affinity is quite weak. And that is important for the reduced cytokine release syndrome risk. So, but with this low CD3 affinity, the monotherapy efficacy is almost zero. You have to combine with 4-1BB, CD137 signaling. So if you are to develop these two molecules in combination, one molecule monotherapy does not have efficacy. So it's quite hard to develop these two molecules in combination in the clinical setting. So that's our thinking, yeah.
So, yeah, I have the microphone. I just had a follow-up question on the 4-1BB, because with the high affinity of CD137, you effectively also made an NK cell redirecting antibody, right? So how is the balance between T cells and NK cells in the tumor model in terms of efficacy of the 4-1BB redirection?
Sorry, I don't have answers. So we have not checked the effect of our antibody to NK cells, so sorry, no answers.
A question more to the tumor target, because you talked about, you started the talk to saying, you know, the CD3 antibodies don't work that well if there's no T cells infiltrating in the tumor. So how much does the tumor target, the DLL3, influence actually infiltration of the T cells in the tumor? Because perhaps endothelial cells, macrophages also play a role in the infiltration of the tumor cells.
Sorry, once again.
Sorry?
Once again, please.
So other cells that you know, so there, you know, like endothelial cells and macrophages might also play a role in getting the T cells in the tumor. And DLL3 is known to sort of also influence that.
Yeah. So, so in this study, we checked several cytokines and also some chemokines, and we see higher level of CXCL9 and 10 production for dual IG. So with this, you know, these cytokines are important for, you know, T cell migration into the tumor. So we think that this increased level of number of T cells is probably due to the higher level of this chemokine production. So we have not checked any like effect on the endothelial cells or macrophage.
Thank you.
So, no, thank you for providing a very elegant presentation in engineering. I have a question with regards to the dual IgG platform, where you define the interacting and non-interacting sites. Do you ever see any incidents of like induced fit where it's not so clear to define what is interacting and non-interacting, or maybe crosstalk of conformational changes?
Induced fit. So this CD3 conformation is changed, but probably this is not like an induced fit. It's just adjusting to the CD137 shape. So sorry, no answer to the induced fit. So if you look at the overall structure, the only change is this in HSCD3. Others are completely, same. But each residue is facing different ways.
Yeah, great talk. Can you comment on target engagement? What's the FDA say when you got 2 or 3 targets? What's the FDA say about measuring target engagement in people?
For this dual IG.
Well, for anything that's more than
Yeah,
two targets.
Yes. Yeah. So, yeah, we had a discussion with FDA so this is now in phase 1 in US. So, yeah. So actually, the binding affinity of the no. So in terms of T cell activation, so actually the CD3 signaling occurs at the lower dosage, and then at the more higher dosage, we can observe additional CD137 signaling. So we explained this to FDA, and they agreed that we start with this dose level, but we want to see the target increase the dosage up to the dose where both CD3 and CD137 target engagement occurs. Is this answering to your question? Yeah.
So finally, what about a gluten-free diet? Do you recommend that for everybody?
Yeah. So for this the gluten and the celiac disease antibody, it's impossible to measure all the.
Okay, I think we're going to take a 20-minute coffee break and then we'll resume.